Thank you. All right, we ready to roll? Everybody got their coffee? Yeah, all right. Might need it. All right, so today we're going to talk about enzymes and regulation of enzymatic activities. Basically, we're going to have a three-part lecture on enzymes, and I'm doing the first part. And then Dr. Traore is going to do the next two lectures on. So I'm going to go over the basics of enzymes and how they work, things that affect them, or things you can use to affect them. And then he's going to do the kinetics and enzyme inhibition in the next two lectures. So with that said, and there's your learning objectives are right in there. So first of all, what is an enzyme? Enzymes are protein catalysts, usually proteins. Now there are some exceptions. There are enzymes which are RNA, and actually maybe even the very first enzymes to ever evolve were, at least some people think, were probably RNA. But usually when we think of enzymes, we're talking about proteins. They're catalysts, so they're going to catalyze a reaction, and I'm going to explain what that means in a second. So they increase the overall rate of the reaction without being changed in the process. So at the very end of catalysis, the enzyme reverts to its original state. And in human biochemistry, these enzymes are used to convert substrates. So we're going to use that word a lot today, substrates. Well, you also see the term reactants, but I usually use substrate, which is the beginning compound, into products, which is what it's going to be converted into. And then more so than that, it actually channels them into pathways. So the rest of biochemistry after today, we're going to be talking about pathways. Okay, so there's not many pathways that we call a pathway that has a single enzyme. So usually we're talking about multiple enzymes. And therefore they direct metabolic events. Now, it's important to recognize that enzymes do not invent new reactions. They don't catalyze something that would not take place normally. It's just they speed it up. Okay, they increase the rate dramatically. So any reaction that's catalyzed by an enzyme, if the substrates were there and you waited long enough, that reaction would occur. It's just enzymes make it happen much, much quicker. And we'll talk about the orders of magnitude quicker in shortly. And so without enzymes, most of the reactions we're talking about would occur far too slowly for life to actually exist. Okay? And by having these pathways also, you can affect the direction of the enzymatic reactions, as we're going to see. Now we're going to talk real briefly about nomenclature, and I don't want you to get too wrapped up in this. There's a couple key points I want you to get out of this. I'm going to tell you right now, I don't want you to try to memorize this figure. This is an FYI. These 6 classes of enzymes are basically all enzymes can fit into one of these 6 classes. But I don't think it's useful to try to make you memorize those. But I think it's useful for you guys to just take a look at that figure. And all those, let me see, we probably won't look at the conversion of serine to glycine, but all of those other reactions we're going to see at some point during biochemistry. Okay, so they're not like real obscure reactions. They're high-yield reactions that we'll see later on. But you don't have to memorize the particular type of reaction. But it's kind of interesting to look and see what's going on. And then as you go through biochemistry, you're going to see things repeated over and over and over again. And it actually will help you learn things and memorize things, because sometimes the enzyme names actually tell you what they do. So first of all, this ASE is usually at the end of most enzyme names. So when you see that ASE, you automatically, if it's something you had never seen before and you see ASE at the end, you're like, okay, that's an enzyme of some sort. Okay, let's figure out what it does. And sometimes it's as simple as it's being added to a substrate of the reaction, like urease. Glucosidase, okay, those are examples you will see later on in biochemistry, or sometimes to a description of the reaction performed, and I think we have that one there, lactate dehydrogenase. Okay, it actually tells you, well, lactate is the substrate, dehydrogenase tells you what's going to happen during the reaction. Probably about, I've never actually added them up, but I would say, 80% of the enzymes we're going to see are of this nature, which makes it very convenient because it sort of tells you exactly what it does. Unfortunately, there's that 20% nagging that you can't really tell, or you can't be certain what it does just from the name. So sometimes enzymes have a trivial name that they were named originally many years ago. A couple we're going to see, trypsin and pepsin. Those are digestive enzymes in the gastrointestinal system. By looking at that enzyme, it does not tell me what it does. I can't, just by reading that, I can't say, okay, I know what reaction it actually catalyzes. So unfortunately, there are enzymes that have names like that. And unfortunately, you're also going to see enzymes which, like for instance, when we get into glycolysis, when we're looking at the pathway of glycolysis, you'll find enzymes that are apparently named backwards. And when I get to that, I'm going to explain to you why they're named backwards. But that can be a little bit confusing as well. So they are divided into 6 classes. Once again, you don't have to memorize those 6 classes, but you're going to see these types of reactions over and over and over again. There is a systemic name that for every enzyme. So those of you who were really into enzymology before you came here, or have a special interest in enzymology, there is a systemic name for all enzymes, and they're generally a complete description of what it does. So they're very informative. If you see that name, you like, okay, I can tell you exactly what's going on in that reaction. But they're also extremely cumbersome, and so therefore they're very rarely encountered in biochemistry texts, in medical texts, and things like that. So unless you're really an enzymologist, you're probably not going to ever use that name. But that's an FYI. I certainly wouldn't expect you to try to memorize this name, but see, lactate dehydrogenase could also be called lactate NAD+ oxidoreductase. So lactate dehydrogenase is easier. And there's some enzymes where it's dramatically easier to say its common name. So the common names are what you're going to encounter. If you do encounter those systemic names, that's what it means. And so really, that's all I really want you to get out of the nomenclature. And as we go through the rest of biochemistry this semester, and maybe you've already had substantial biochemistry before and you've already noticed this, you're going to see patterns repeating. Those patterns sometimes are going to be really useful for you guys to try to remember the enzymes. Unfortunately, when you go to take your board exams, you're going to have, they're going to describe a patient, and you're going to have, if it's a biochemistry question, it's probably going to be, there's a good chance that it's going to be enzyme choices. One thing you need to be careful of is the enzymes start to sound the same. You're going to find a whole bunch of enzymes that start with glucose. Okay, glucose 6-phosphatase, glucose 6-phosphate dehydrogenase. Wow, those sound the same, but they do completely different reactions, and they're important for completely different reasons. So when you encounter those, take special care. That might be one of those situations where when you start encountering things that sound sort of the same, where you want to actually use a flashcard, for example, to study from. Another key thing when you're looking at figures, and you can actually see it in this figure here, you'll see there's arrows, okay? And you'll notice in some of these reactions, the arrows are going in both directions. And in some of the reactions, the arrows are only going in one direction, okay? That's actually important. Now, I'll warn you ahead of time that not all texts follow this rule, so you have to be a little bit careful, but for the most part, they do. If you see the reaction, the arrow going in both directions, that means that enzyme, under physiological conditions, can catalyze the reaction in either direction. So for example here, lactate dehydrogenase catalyze the conversion of lactate to pyruvate or pyruvate to lactate. And you'll see actually in the body it does both of those. And it's important to do both of those reactions. And it depends on what cell type and what conditions are going on, which way it's going to go. Whereas other enzymes can only catalyze the reaction under physiological circumstances in one direction. Urease is an example here. I don't think we're going to see urease in biochemistry. You may see it in later on in systems, though. But pyruvate decarboxylase would probably see that enzyme later on. But you're going to encounter numerous, and actually the most important enzymes in biochemistry, the ones that control pathways, usually the arrow only goes in one direction. Once again, that means under physiological conditions, the conditions that enzyme would normally encounter, it can only catalyze in one direction. Now, the reality is, under artificial circumstances, you can make an enzyme, any enzyme, go backwards. But under physiological conditions, some enzymes, you can never get the conditions to such an extreme state to make it go backwards. Okay? So, and once again, most of the real important reactions that we're going to spend a lot of time talking about, the rate-limiting pathway or enzymes for pathways, usually they only go in one direction. Okay, so we're going to look at the properties of enzymes now, and just to remind you, enzymes, as we're going to talk about them today, are protein catalysts. They're going to increase the velocity of the reaction, and they're not consumed. And they may undergo a temporary change, but at the end of the reaction, they're converted back to their original state. So some of the properties of the enzymes, they all have an active site. Okay, so here's some terminology I want you to know here. The active site is going to be, and in this case, it's a real convenient visualization. It's going to be this little cleft that's going to bind the substrate. Okay, so you're going to have some interaction with that substrate to catalyze a reaction. And so it's going to have a complementary 3D surface. It allows amino acids within that enzyme to interact with the chemical groups on the substrate. Okay. This is going to be important because we're going to talk about some of the things that can affect enzymes shortly. And if you imagine this complementary 3D and the amino acids within the enzyme being important for catalysis, then you can start to think of what some of the things that might actually affect that are. And then, so, if you bind it, you're going to, and this is terminology that you're really going to see much more in the next two lectures. You get this enzyme substrate complex, gets converted into enzyme product, and then the product is released and the enzyme goes back to its original state. That's all this is talking about. And that's, once again, that's going to be important in the next lecture. Catalytic efficiency. So enzymes are very efficient. So somewhere between 1,000, and if you calculate what 10 to the 14th is, so it's 100 trillion times faster than uncatalyzed reactions. Okay, so it's very, very, very significant. The turnover number, so this is the molecules of substrate per converted to product per second. So that's on the order of 100 to 10,000. That boggles my mind. I can't visualize when I'm thinking about an enzyme working and substrate coming in and binds and then it goes a conformational change and lets it. I can't imagine that happening 10,000 times in 1 second, but it can. So it's pretty amazing. So very, very efficient, and they really speed up the whole process. Enzymes are also very specific. So they work usually only on a single substrate. So you're going to have, that's why you have so many different enzymes. So for every different substrate, you need a different enzyme. There are some enzymes that have a few different substrates, but the substrates are very closely related. Okay, for instance, in your body, alcohol dehydrogenase can work on ethanol and methanol. Actually, can work on some other chemicals too that have an alcohol group. That's just an example, but all of those have very similar chemical properties and can interact with the enzyme similarly. But most of them really only have one specific substrate, and they only catalyze one type of reaction. Okay? So if you do see an enzyme that has multiple activities that, like we already talked about, like some DNA polymerases that did, they did multiple things. They have multiple subunits, okay? And so there's different enzymes on different subunits. And then we have this concept of holoenzymes. Now, I know we've already used holoenzyme when we were talking about the prokaryotic RNA polymerase. And unfortunately, it's a little bit confusing because this is the more conventional definition of a holoenzyme. In that case, the extra factor that needed to be added to make a holoenzyme was a protein component. But more traditionally, the usual definition of holoenzymes are enzymes, and the enzyme itself would be the apoenzyme, which would be inactive, that have some kind of non-protein component, okay? And you need that non-protein component to make the holoenzyme, and the holoenzyme is actually the active enzyme, okay, in this case. If that component happens to be a metal ion, zinc, iron, magnesium, so some type of metal ion, that's going to be called a cofactor. So that holoenzyme, or that apoenzyme, requires a cofactor, this metal ion, to make the holoenzyme. If the component is actually a small organic molecule, it's called a coenzyme. Okay? And coenzymes are very often derived from vitamins. So a couple examples we're going to see over and over and over and over again in biochemistry. NAD+ is derived from niacin, FAD derived from riboflavin, but most of your water-soluble vitamins, at least one of the mechanisms in which they can work is as a coenzyme for certain enzymes, okay? So once again, a coenzyme is a small organic molecule, very often derived from vitamins, whereas a cofactor is a metal ion. Enzymes can also be regulated, and we're going to talk a little bit about regulation, so we can actually activate or inhibit them depending on the needs of the cell. So that allows us to turn on or turn off certain pathways. Okay, and that really gets to the core of metabolism. And in eukaryotic cells, you guys know there's all these discrete areas, organelles, and things of that nature. So the enzymes are located in specific subcellular compartments. That can isolate substrates and products from competing reactions. So as we go through biochemistry, you're going to see that, for example, when glucose comes into a cell, there's multiple different things that can happen to glucose. Some of those things can be controlled by turning enzymes on and off, but you can also control things by compartmentalizing where glycogen synthesis is going to take place, or something of that nature, some of the other pathways. So when you have competing substrates, you can't just have enzymes just going at random and trying to all be on at the same time, because things would never work that way. So part of the whole organization is going to be isolating things. And even within bacteria cells, you can get discrete granules within the cell where a bunch of enzymes involved in the same pathway are located together to kind of shuttle the substrates through and isolate it from something else. Okay, so how do enzymes actually work? So there's two perspectives. These aren't mutually exclusive. It's just two different ways of trying to visualize the same thing, or try to visualize what catalysis actually means. The first is, or one way of visualizing it is thinking about energy changes during the reaction. So you have this energy barrier. So what is the energy barrier? It's the difference the energy difference between that of the reactants or the substrate or substrates, depending on what reaction we're talking about, it's some high energy intermediate that occurs during the formation. So if you look at this figure, A would be the substrate or substrates, B would be the product or products. Okay, and if you look, you'll see that B actually has a lower energy at the end. So that should be a spontaneous reaction. But you actually, in order for that to happen, you actually have to get over this hump right here. And that hump is the free energy of activation. Okay? So one of the ways of visualizing what an enzyme actually does is it actually lessens this hump, this free energy of activation. So it lowers the free energy of activation. So in this figure, this blue line is showing you the uncatalyzed reaction. Okay, if there was no enzyme there. If you give it long enough, that will actually happen. But what an enzyme actually does is lower that hump. So this red line is actually showing you a catalyzed reaction. It lowers that hump, so it makes it more favorable for that reaction to actually occur. Okay? Now you'll notice we didn't actually change the free energy of the product or the reactants, the substrates. Okay, they're both the substrate starts at the same spot and the product ends at the same spot. Okay, so you just changed that free energy of activation. So that's one way of visualizing or conceptualizing how an enzyme works. Another way is to think about the chemistry of the active site. So the enzymes can actually stabilize the transition state. So in this figure, the transition state would be that high energy intermediate. So once again, these two aren't mutually exclusive. They're just two different ways of visualizing the same thing. And it does that by holding the substrate in some kind of high energy conformation, kind of the peak of that hump. It holds it there so that it can actually go over that hump. So if you hold it there long enough, chances are it's eventually going to go over that hump and move forward in the reaction towards the product. So that increases the concentration of the reactive intermediate, therefore increases the amount that can go into the product. Now, you guys can definitely see that, we're talking about a line here. It could go the other way, certainly, and if you're holding something in a high energy intermediate, it could go either towards the product or it could go back to the substrate, right? And that'll be important in the next couple lectures to think about as well. Now, there's certain things you can do to actually drive reactions forward, and we'll see that when we actually start talking about pathways. And so what ends up actually happening, and your textbook actually uses this, this idea of trying to take a sweater off a little kid who's not necessarily being very cooperative. And if you can get him in that high energy intermediate, which is kind of with his hands raised up, and you could actually put the sweater back on, or you could pull it off, right? But getting to that high energy intermediate is the trick, and that's what the enzyme will actually do. And so you have these chemical groups within the enzyme that are actually going to interact with that substrate and actually hold it in some kind of high energy conformation. Then once again, at the end, the enzyme is going to be returned to its unaltered state, so it'll go back to normal. Okay, so next we're going to look at factors affecting reaction velocity. It really, when I say the reaction velocity, I'm talking about the initial reaction velocity. So that's that V sub 0, and that's, I probably should have made a bigger deal out of this right here, getting that straight in your head, or I really probably should have used it in the title and all. I was running out of room going across. So it's initial reaction velocity, and I think Dr. Traore will spend a couple minutes explaining to you why it's important to look at initial reaction velocity. Once things start going and you start accumulating product, that's going to change things because you can actually go in the opposite direction. So you're always looking at initial reaction velocity. By the way, does this graph, not reading the particulars of the graph, but does that look familiar to you? Looks like hemoglobin and myoglobin, doesn't it? If you've got a good grasp on what's going on with hemoglobin and myoglobin in terms of oxygen binding and release, that's exactly how enzymes work. Now, hemoglobin and myoglobin aren't enzymes, but they're almost the perfect model to show how enzymes work. So your myoglobin actually looks like that blue line. And then what was going on with oxygen that made this happen, this sigmoidal curve? You had that cooperativity, right, where binding of an oxygen molecule affected further binding of other oxygen molecules. Well, those are what allosteric enzymes look like, and we're going to spend some time talking about that. Most of our super interesting enzymes in biochemistry actually follow this green line. So the velocity is the number of substrate molecules per time, okay? And really, once again, we're always looking at the initial velocity. Once product starts accumulating, it affects the velocity. The maximal velocity is as fast as a given concentration of enzyme under the given conditions can go. So you keep everything, the conditions all the same amount of enzyme, the same temperature, pH, things like that. And then at a certain concentration of substrate, meaning eventually you're going to saturate the enzyme. Say you had 100 molecules of enzyme and you just keep dumping substrate. Eventually you'll saturate all the active sites and it can't go any faster. That is your Vmax. So once again, you'll see these two types of curves. Most enzymes show this Michaelis-Menten kinetics, which is this blue line, real simple. But the real interesting enzymes, the ones that are going to control pathways, usually show this green line, which is our sigmoidal curve. Okay, so what actually can affect reaction velocity? First thing we're going to look at is, I should have said the first thing that can affect reaction velocity is the concentration of substrate. So given a concentration of enzyme in identical conditions, the more substrate you add, the faster it's going to go until you saturate it. Okay, and that when you saturate it, you're at your Vmax. Okay? However, temperature can also affect the reaction velocity. And what you'll get is if you plot initial reaction velocity, so keeping everything else the same except temperature, if you plot over temperature, you're going to see this type of bell-shaped curve where the reaction velocity is going to peak at a very specific time, or a very specific temperature. So, generally speaking, for enzymes, the reaction velocity, if you increase the temperature, the reaction velocity is going to go up. Okay? That's true for all enzymes until you reach a certain point. Okay? And at a certain point, the reaction velocity now is going to start to go down. So that point is where you start to actually denature the enzyme. So all enzymes, until you get to the point of denaturing them, they always go faster if you increase the temperature. Okay? If you increase the temperature, the substrates have more free energy, so it's easier for them to get converted into a product. Okay? So and that also will increase the rate of the reaction without an enzyme as well. But once you get to a certain temperature, you're going to start to denature the enzyme. So though you remember all those different types of bonds we talked about that make the 3D structure of a protein. If you increase the temperature, some of those hydrogen bonds and ionic bonds are going to start to break, and you start affecting the 3D structure of the protein, and that's going to be important when we talked about the active site and how it interacts with a substrate. And eventually you will destroy the enzyme if you get the temperature too high. Human enzymes have a temperature optimum around 37, which happens to be body temperature. Okay, and if you get much beyond about 40 degrees centigrade, that's when things start to denature. And this is why high fevers are potentially life-threatening. You can actually drop somebody's body temperature down pretty significantly without killing them. Now, you can't do it for a long period of time, but and actually some types of surgery, they'll even try to do that. But you can, and you'll slow their metabolism way, way, way down, but they'll still be alive. But people cannot survive a very high fever for much longer than a short period of time, okay? Because it's going to start to denature those enzymes. And in many cases, enzymes, just by lowering the temperature, won't always go back to their correct structure. So sometimes when you denature things, they're permanently denatured. And if you denature the right things, there's no way for you to go back and make more, right? You start denaturing the things involved in protein synthesis. So this is why high fevers are extremely dangerous, okay? Why they try, if somebody does have it, they'll go into emergent situation where they're gonna, like, if it gets high enough, they're gonna, like, dump them in an ice bath or something, try to get that temperature down any way possible before it kills them. pH will also affect reaction velocity. Now, what you'll notice here, though, is that unlike with temperature, when we're talking about humans, almost all human enzymes have a temperature optima around body temperature, but they can have very different pH optima, and that depends on where these enzymes are actually going to be working. Okay, if you have an enzyme that's going to be working in the low pH of the stomach, like pepsin, for instance, it would have a very low pH optima. Most of your enzymes are working in an environment that's, we have a pH somewhere around physiological pH, which is around neutrality. It's a little bit higher. It's about 7, 4 in that range. But I always just say 7, just to say, physiological pH. Most of the enzymes are in that range. But if they have a special compartment, like lysosome, for example, stomach or something, they may have a different pH optimum. And so why is that, or what's going on? Well, just like when we're thinking about temperature, you have all these interactions going on that create the 3D structure of a protein. And if you alter the pH, then you start changing whether certain groups within that structure are protonated or unprotonated. So you start changing those hydrogen bonds and those ionic bonds, right? And then you alter the structure. So, therefore, each enzyme is going to have a certain pH optima. If you get too far outside of that range, you really start affecting its activity. If you get far enough outside of the range, you're going to completely denature that enzyme and render it inactive. Okay, so next we're going to look at the regulation of enzyme activity. So the rate of enzymatic reactions in the body is very responsive to changes in substrate concentration. And that's because the normal physiological amounts of substrates that you would actually see is going to be near the range of the Km. Now, have you guys seen Km yet? You have seen Km. Awesome. Cool. Well, you're going to see it a lot in the next 2 lectures. I was hoping I didn't have to explain it. So you guys know what Km is, right? It's basically affinity, so if you look at, let's just jump forward here real quick. You'll see where the Km is. All that's saying is between 0 and where the Km is, if you use your imagination, it's not a perfectly straight line, but it's roughly a straight line, right? So that means, and what's happening here is we're increasing the substrate concentration. So as you increase the substrate concentration, the enzyme goes faster. And usually, under normal circumstances, you don't get out to that Vmax where you're actually going to saturate your enzymes. So usually they're working within that physiological range. And so that's one way we actually regulate the enzymatic activity. If a whole bunch of substrate's there, it's going to turn that enzyme on, or that enzyme's going to be active. And if there's not a lot of substrate there, it's not going to be active. Okay? And so the bulk of the enzymes are going to actually be primarily regulated that way. They don't have any kind of special regulation Those aren't the ones we're going to spend a lot of time on in biochemistry, but you'll see, for example, next week I'm going to talk to you about glycolysis. You're going to see a bunch in glycolysis that just, which way they go and whether they're on or not just depends on substrate. Now, some enzymes can respond to allosteric effectors and or covalent modification. Okay, and that's going to be the enzymes are really going to be concerned with. So we have things that are going to, beyond just the substrate concentration, we have things that are going to turn the enzymes on or off. Rate-limiting enzymes, as we're going to see, and we've already alluded to during gene expression, can actually also be regulated by the amount there. We can actually make more or make less of an enzyme to regulate a pathway as well. So the regulation of allosteric enzymes, so these enzymes have a velocity plot here, which would normally call that a Michaelis-Menten plot. They will show that sigmoidal curve, kind of like oxygen binding. So that means there's something else there affecting the speed of the reaction. So allosteric enzymes are regulated by some kind of effectors that bind to sites other than the active site. So we're talking about some other spot. Very often these allosteric enzymes actually have multiple subunits, and where the allosteric effector actually binds is a different subunit. When I say a different subunit, I mean a different polypeptide. This is 2 proteins, 3 proteins, 4 proteins, multiple proteins coming together to make the active enzyme. Very often the allosteric effector is binding somewhere on a different protein component than where the enzymatic activity is. The effectors can alter the affinity, so the Km. They can also alter the Vmax, so they could alter either or both. Okay, and it could be a positive or a negative regulation, as we're going to see. The effectors that inhibit the enzyme activity are called negative effectors, but decrease the Vmax or increase the Km. You guys said you already saw Km, right? So you know an increased Km means a lower affinity, right? Somebody say, yeah, you already knew that. Okay, good. If you have an effector that increases enzyme activity, that would be a positive effector, so it would make a higher Vmax, so it would increase Vmax, or decrease Km, which means it's actually increasing the affinity. These effectors could be homotropic effectors. This is when the substrate itself serves as an effector, and most often a positive effector. If you're trying to visualize what's going on there, if you understand what's going on with oxygen binding to hemoglobin, that's what we're talking about. When one molecule of oxygen binds to hemoglobin, it actually makes the second one bind even stronger, right? And so on till you saturate the oxygen molecule. That's what we're talking about here. So in that case, the presence of the substrate in one site increases the catalytic properties, so either increase the Vmax or decrease the Km, which increases the affinity of the other binding sites. And that's where you get this sigmoidal curve. You can also, that's a homotropic effector. You can also have a heterotropic effector, and this is when the effector is different than the substrate. And this is the more common thing to see. For example, you can have feedback inhibition, where some product of a pathway further down a pathway will actually come back and inhibit an enzyme near the beginning of the pathway. And that's when we get to these reactions, you'll see that makes logical sense because you don't want to start this whole pathway if you don't need any more of the product that you're trying to make. So you want to stop it at the beginning and then convert that substrate into something else. Okay, and that's the more common thing, and that's the thing we're going to see endless amounts of time in the rest of biochemistry. I've given you an example here, phosphofructokinase 1. If this is an enzyme you haven't seen before, PFK1, you will be intimately familiar with it next week. So, but it's a good example of one where There's a product that comes later on, and actually it's a product that's an intermediate of the TCA cycle, citrate, that will actually come back and inhibit that, because you don't want to go past that step if you don't need to go down that pathway, because you could convert glucose into something else. In this case, for example, glycogen. And we'll see that next week. You can also regulate enzymes by covalent modification. We've alluded to this a little bit earlier. You can turn enzymes on or off by phosphorylating them, for example. Now, we talked about the enzymes kinases, phosphorylate, phosphatases, dephosphorylate. The amino acid residues are ones which have hydroxyl groups, right? We already talked about those things. Now, unfortunately, there's no way to know, not given anything else, just by seeing an enzyme, there's no way to know whether it's going to be active or inactive by being phosphorylated. Okay, some enzymes are activated by being phosphorylated, some are inactivated by being phosphorylated. There is, within metabolism, there's this general rule, fasting is phosphorylation. So in metabolism, and I know you haven't seen the introduction to metabolism yet, you basically got two ideas. One is you're in the fasting state, one is in the well-fed state. So either you've just eaten and you have a lot of things available, or your last meal was a while ago, and so the most dramatic differences in metabolism is where you're going to see is between those two things. So if you've just eaten, your body wants to make a whole bunch of things, and if you haven't just eaten, then your body wants to break down things. Okay, and in order to do that, it turns on a whole bunch of pathways and turns off a whole bunch of pathways. And they're kind of, the pathways are kind of opposite in direction. And when you're fasting, you phosphorylate all these enzymes. Now, some things get turned on, some things get turned off. Eventually, you'll be able to figure out what those are. And then you have exactly glucagon is your fasting hormone. Insulin is your well-fed hormone, indicating you just ate something. And insulin will actually dephosphorylate all those key enzymes. Once again, some of those, it turns on some of those enzymes, it turns off other ones. So right now, there's no way for you to know. Once we start getting into the pathways, you'll figure out which pathway is going to be active during fasting and which one's going to be active during the well-fed state, and you'll be able to figure out, okay, phosphorylating is going to turn on this particular enzyme and turn off this one. And you can also affect enzymatic activity ultimately by induction or repression of enzyme synthesis. So we're talking about gene expression here. So you can control the amount of mRNA and the amount of protein being produced from that mRNA in various ways, which we've already talked about. You could also potentially increase or decrease the degradation rate of either the mRNA or the protein itself. Okay? Usually the enzymes subject to this type of regulation are only needed under specific physiological conditions. So these would be enzymes that are not in constant use. If you have an enzyme that's required all the time and in relative abundance all the time, it's just going to be made all the time, and you're not going to be turning that on or off. It's just going to be, that's what, you can kind of think of that as when we talked about our housekeeping genes. If you have enzymes that are only going to be on at certain times, or the pathway could be, it might be on all the time, but it's not needed as much under certain conditions, for example. So then you want to temper back, or it may be needed even more in certain situations. You want to actually increase the expression. A good example is some of the specific enzymes that are unique to glycolysis. Now you'll see when we talk about glycolysis, there's a bunch of enzymes you can use either for glycolysis or gluconeogenesis, which go in opposite directions, and they would be on at different times. So I'm talking about the liver here right now. The housekeeping ones, the ones that go in both directions that are used for both pathways, would be around all the time. But the ones that only go in one direction, like glycolysis, for example, they would be under this type of regulation. So if you had a high carb diet and you've got a lot of glucose and your liver's dealing with a lot of glucose all the time, it would respond by making more of the enzymes involved in that. Conversely, if you hardly ever ate any carbs and you really never were doing glycolysis in the liver, you would have very few of those enzymes available. Okay? Now, if you're going to actually regulate an enzyme that way, this isn't something instantaneous. You could really think of the things like if you phosphorylate or dephosphorylate an enzyme, you do that signal transduction pathway, you turn it on or off, that can happen in seconds. If you're talking about changing gene expression, you're talking about hours to days to really see that effect. And that's why, you know, the steroids we've talked about, all steroids work by binding to an intracellular receptor, a receptor in the cell and affecting gene expression. So it always takes a little while before you actually see the effects. It's not immediate. Okay, and so the last topic for today is going to be some basics of enzymes used in clinical diagnosis. So there are some enzymes that are present in the blood and are active in the blood and are supposed to be in the blood, like, for example, enzymes involved in blood coagulation. However, there's a bunch of enzymes that end up in the blood through the lysis of cells. So cells get turned over, they break open, and the enzymes get released, and they end up in the blood. And so you'll have a certain level of those enzymes around at all times. Sometimes it's detectable, sometimes it's not detectable, depending on how sensitive the test we have is. But there's always some presence, and that's during normal cell turnover. But that amount is always going to be small and constant. And so you're eventually going to release, you're actually going to pull those enzymes out and actually degrade them. However, there are enzymes that when you have some type of tissue damage, so you somehow damage tissue through trauma or whatnot, you're going to break open the cells, and these enzymes are going to be released into the bloodstream, and they increase the concentration. So we can actually use that as a diagnostic tool, and it can be a diagnostic tool that can tell you, by looking at specific enzymes, what tissue got damaged, or in some cases, how extensive the damage might actually be. Okay? And so, once again, the level of the enzyme could be correlated to the extent of the damage, so it can help you determine the prognosis for the patient. So, how does that actually work? Well, within different tissues are going to have different pathways active, or in some cases I'm going to talk shortly about, they might have isoenzymes, so they might have the same pathway active, but the enzymes are a little bit different, and you can distinguish between them. But some tissues have particular activities that are unique to that tissue, or are maybe not unique to the tissue, but are in much higher abundance in a certain tissue. For example, the enzyme alanine aminotransferase, particularly abundant in the liver. So you're going to see a lot of that in the liver. If you get very high levels of ALT in the bloodstream, that's indicative of liver damage. Now, ALT is just going to be one of many enzymes that you would look at in a liver function panel. So there's multiple other ones, because you could have ALT possibly elevated through some other damage as well. But you can get a total picture by looking at a bunch of different enzymes that would be more or less, not necessarily unique to the liver, but in much higher abundance in the liver. So you can actually determine liver damage. And you can do that with a bunch of different tissues. And one specific one we're going to look at today is isoenzymes in diseases of the heart. An isoenzyme catalyzes the same reaction, but it's different at the amino acid level. That means we got two different genes, or maybe more than two, but at least two different genes encoding a similar protein. It's similar in terms of its enzymatic activity, but it differs in the amino acid sequence. Therefore, there's ways you can differentiate it. Could be electrophoresis, like what they're showing here, or could be another way. And different organs can frequently contain characteristic populations of these isoenzymes. Some of these isoenzymes actually have different subunits in various combinations, which is what we're going to see right here with creatine kinase. Okay, so there's actually two different forms of this creatine kinase, the B and the M form, and it always works as a dimer. So therefore, that means you have three possible combinations: BB, MM, or MB. And you can actually take advantage of this to diagnose an MI, a myocardial infarction. Okay. Now, I actually told a little bit of a lie here. BB is not found only in the brain, but it kind of simplifies the model. Okay, you'll see a little bit of BB in other tissues as well, but the overall story is basically BB is the isoform you see in the brain, MM is the isoform you see in your skeletal muscle, and in the heart, you'll actually see both MM and MB. So really, the only place you see a significant amount of MB, or this CK2, and I'd like you to know both names, CK2 and MB, is going to be in the heart. Therefore, if you see an elevated level of MB in a serum sample, it's indicative of a myocardial infarction. And the relative amount can tell you the severity of it. So here's about 4 to 8 hours after MI, peaks at about 24, and after about 48 to 72 hours, it's going to return to baseline, which you can see in this green line in this graph. I think my microphone's going out. Sorry. I'm trying to finish, so sorry. You can come up and ask me after class. So that's one way we can actually diagnose myocardial infarction. You also have troponins, which are used and considered a newer test in the gold standard, although they're actually revising this creatine kinase, so they've actually made it a little bit better now, so they're starting to use it again. But there's multiple different ways, and so here's another isoenzyme, troponin I, is found primarily in the heart. So it's very specific. If you see this elevated in the plasma, it's going to be indicative of cardiovascular or heart damage. So it's going to be indicative of an MI. And because it actually gets to a much higher level, it's easier to detect. It gets to that high level as quick as CK, but it actually remains elevated where we can actually detect it for a much longer period of time. So very often with MIs, the patient didn't know they were having an MI and don't come in until days later. And sometimes, and so that test can be valuable to diagnose, if somebody had an event 4 or 5 days ago, for example. Okay. And once again, I included your little figure there for you guys to use to study from. Sorry I ran a little bit over. If you want to come up and ask me your question, I can answer it. Sorry I didn't get to do it during class.